labs to gather important samples from those raccoons. They'll take a blood sample, they'll take a tooth sample, and then some very basic data, age, weight, and sex of the animal. Typically what happens once we have the raccoon immobilized is that a blood sample is taken. The blood sample is taken so that we can do a serum test, essentially, to see whether the raccoon has been exposed to the vaccine and has created antibodies to rabies. In addition, a tooth sample is also taken, and that gives us two pieces of information. Number one, it can help us age the animal, and basically the tooth in the lab gets cross-sectioned and we can count the rings of the tooth. We also are looking for the presence of a tetracycline biomarker that's actually in the bait, and that allows us in the lab to tell whether that raccoon has had contact with that bait. Once we've taken the samples from the raccoon's allowed to recover, and then it's released back into the wild. Roadkill surveys are also an important component of our oral rabies vaccination program. On the one hand, we can post-bait look to see if these roadkills are positive for the tetracycline biomarker, indicating they've been exposed to the bait. And we can also, if it's a fresh specimen, test to see if they're positive for rabies, helping in our overall rabies surveillance program. Similar to the oral rabies vaccination program in raccoons that we're doing in the eastern United States, we're working with cooperators in the western United States and Texas that has an oral rabies vaccination program that focuses on gray fox and coyote. Now these programs in gray fox and coyote are very similar operationally to the raccoon program in the eastern United States in terms of how we distribute the bait as well as the type of bait and the vaccine that we're using.
In approximately 1988, a variant or type of rabies called domestic dog coyote variant became epizootic, and that's a term that describes an epidemic in animals. And that occurred in South Texas on the county bordering our international border with Mexico. That variant became very widespread in South Texas and was moving toward major metropolitan areas, particularly San Antonio, at a rate of about 40 to 45 miles per year.
If the canine rabies, which moves through coyotes, it could move all the way through North Texas into other states of the United States. The other big concern in South Texas was with the coyotes that it moved the rabies in South Texas, but then they would interact with unvaccinated dogs, and that made it one of the bigger health threats because unvaccinated dogs then would go home and cause real human health and safety issues.
The Texas ORVP was developed and implemented beginning in 1995. It's been very successful. We went from 1994, the year previous to the actual implementation, there were 166 cases of that type of rabies in Texas. In 2000 there were none, 2001 there was one, and we haven't had any since. So all indications are that that variant has been eliminated from the United States. But we maintain a barrier of bait dropping along that international border to maintain a population of immune coyotes so that we don't get a reincursion from Mexico, because that variant still exists in Mexico. So we collect specimens after the bait drop and submit samples of maxillary bone and teeth to detect the presence of tetracycline. And if it's present, it suggests then that that animal has consumed bait. In fact, in a growing animal, we can tell how many they consumed, and in adults, you can tell over how many years they've consumed bait. We also do serology or serum testing to determine what percentage of the animals have developed antibodies to rabies. For example, in 2003, we've recently got our results back, and in coyotes, 77%, had shown presence of antibody to rabies. If you're going to break the cycle of a disease in a population, whether it's a human population or an animal population, you have to have a certain level of resistance. And in this case, that resistance is brought about by vaccination. So that if a rabid animal contacts an animal that's protected against rabies, that animal will not, the animal exposed will not break with rabies, and the cycle is more readily broken and dies out in nature.
It's of great interest to us, and we've begun some initiatives with Mexico to enhance border rabies surveillance to ensure that our programs are of the proper integrity along the Texas border to keep coyote rabies from becoming reestablished in Texas and the United States.
Skunk rabies is the next major obstacle as far as coming up with both vaccine and vaccine bait delivery systems to deal with skunk rabies, which is epizootic throughout the United States. And so even those states who do not have coyote rabies, which is none at this point, or gray fox rabies, or raccoon rabies, they do have, many of them have skunk rabies.
Skunks are the second most common animal to have rabies in the United States. They're a very important rabies virus reservoir. So, and the vaccine that works for raccoons does not work orally in skunks. So we've been doing quite a bit of evaluation of new types of vaccines that may control rabies in skunks, because they're obviously the second most common, the second most important species for which to develop a vaccine.
We are discussing looking at the economics of, is it worthwhile to develop new technologies along the lines of the current oral rabies vaccine for raccoons, but for skunks and indeed other animal species, with skunk rabies in California being a particularly menacing problem in the future, and we'd like to develop it. And then also developing new ways to deliver it, finding new means for presenting it to the animals, new types of baits, the technologies. This is all very formative, but it's crucial to understand at the early stages what these new technologies mean in terms of the economic cost of delivering it.
The most unique species that we have are probably the big brown bats. They're very important because even if we could solve terrestrial animal rabies, rabies in skunks, rabies in raccoons, we still have this problem with rabies in bats, and it's very complex and quite different from approaches that we would use for our terrestrial animal species. So we're looking at the pathogenesis of rabies virus in those, looking at different aspects of the bat's immune system, and hopefully we'll go on from there to try to find better ways of preventing rabies in those animals.
Their diverse, their numbers, their densities, the kinds of viruses that they have, and the mechanisms that we would use to reach them via aerosols, baits on the wing, etc., just boggle the imagination as to how one could effectively reach free-ranging bats beyond the traditional measures of public health prevention and control.
Hang all right. Yeah, you can hang all right. Should be a stream down here.
Currently, the focus of oral rabies vaccination is on terrestrial carnivores. With a little bit of luck and a lot of funding, one potentially could fill in the blanks from the Great Lakes to the Gulf, from the Appalachians to the Atlantic, and potentially consider eliminating raccoon rabies from the eastern United States.
The program is a very exciting, complex, and challenging program, and we made some very good successes in terms of containing and trying to eliminate strains that we're currently focusing on. However, we cannot become complacent. We need to have better vaccines. We need to continue to work toward better vaccines, better baits, and better ways to deliver them so that we can ultimately achieve our objective of eliminating terrestrial rabies in the United States.